Want to ace your test in just a few minutes? Watch this video. Define microscopy, the use of a microscope to view objects not visible to the naked eye. What is magnification? The increase in the apparent size of an object. What is resolving power? The ability to distinguish between two separate objects. How does a light microscope work? It uses visible light and two lenses to magnify the image of a specimen. What is meant by LM109X? It indicates the photomicrograph was taken with a light microscope and magnified 109 times. Compare light and electron microscopes. Light microscopes magnify up to 1500x, while electron microscopes can magnify up to 250,000x with higher resolution. How does an electron microscope work? It uses a beam of electrons to magnify the specimen in a vacuum. What problem do scientists face using electron microscopes? Electron microscopes cannot study live specimens because they require a vacuum. What is the difference between SEM and TEM? SEM scans surfaces. TEM transmits electrons through specimens to study internal structures. What did Robert Hooke contribute to cell theory? He discovered cells in cork in 1665 and coined the term cell. What was the role of Schleiden and Schwann in cell theory? Schleiden stated that all plants are made of cells. Schwann said the same about animals. What are the postulates of cell theory? All organisms are made of cells. Cells are the basic unit of life. All cells arise from pre-existing cells. Why are acellular particles not included in the 5 kingdoms? They are not made of cells, as viruses, prions, and viroids lack cellular structure. What are the structures in a cell that are not organelles? Cell wall, cell membrane, cytoplasm, and cytoskeleton. What is the difference between primary and secondary cell walls? Primary walls are made of cellulose, while secondary walls have lignin. What is plasmodesmata? Pores in plant cell walls that connect cytoplasm between adjacent cells. What is the role of the cell membrane? It acts as a semi-permeable barrier, controlling what enters and exits the cell. What is the difference between plasma membrane and cell membrane? The plasma membrane is the outer boundary, while cell membrane refers to all membranes in a cell. Draw and label the diagram of the cell membrane. The diagram includes a lipid bilayer, proteins, and glycoproteins with the outside and inside of the cell labeled. What is the fluid mosaic model? A model of the cell membrane with a lipid bilayer in which proteins are embedded. Define cytoplasm, the semi-viscous substance between the cell membrane and the nuclear envelope containing organic and inorganic molecules. What are the functions of the cytoplasm? It provides space for organelles and is the site for metabolic reactions like glycolysis. What is the difference between microtubules and microfilaments? Microtubules are made of tubulin and help in shape maintenance. Microfilaments are made of actin and help in cell movement. What are organelles? Small structures within a cell that perform specific functions. What is the nucleolus? A dark spot within the nucleus where ribosomal RNA is synthesized. What are ribosomes? Tiny granular structures in the cytoplasm or attached to the endoplasmic reticulum, made up of proteins and rRNA. What are the functions of mitochondria? Mitochondria are the powerhouses of the cell, responsible for energy production through aerobic respiration. What is special about mitochondria? Mitochondria have their own DNA and ribosomes, similar to bacterial ribosomes. Define chloroplast. Chloroplasts are organelles in plant cells where photosynthesis occurs, containing chlorophyll and other pigments. Where are chromoplasts located and what are their functions? Chromoplasts are found in flower petals and fruits and are responsible for their bright colors to aid in pollination and seed dispersal. What is the difference between SER and RER? SER lacks ribosomes, synthesizes lipids, while RER has ribosomes and helps in protein synthesis. Who discovered Golgi apparatus? Camillo Golgi discovered it in 1898. What is the function of lysosomes? Lysosomes digest waste and foreign materials. What is a centrosome? A centrosome is a region that helps in cell division. What happens if lysosomes burst? It causes cell death by digesting the cell's own contents. What is the difference between food and contractile vacuole? Food vacuoles digest food, while contractile vacuoles remove excess water from cells. What is a prokaryotic cell? A simple cell without a nucleus, like bacteria. Difference between prokaryotic and eukaryotic cells? Prokaryotic cells lack a nucleus, while eukaryotic cells have a nucleus. How does cell shape relate to its function? Cell shape is adapted for its specific function, e.g., nerve cells are long for communication. What is the open system of a cell? A cell exchanges energy and matter with its environment. How are surface area and volume related to a cell? Cells need a high surface area for efficient exchange of materials. What is the difference between diffusion and facilitated diffusion? Diffusion is movement from high to low concentration, while facilitated diffusion uses proteins. What is osmosis? Osmosis is the movement of water through a semi-permeable membrane. What is tonicity? Tonicity refers to the concentration of solute in a solution. What happens to a plant cell in a hypotonic solution? The plant cell takes in water and becomes turgid. What is turgor pressure? It is the pressure exerted by water inside a plant cell against the cell wall. What is plasmolysis? Plasmolysis is the shrinking of a plant cell's cytoplasm due to water loss. How does osmosis help in stomata opening? Osmosis causes guard cells to swell and open the stomata. What is reverse osmosis? It is the process of removing impurities from water by applying pressure. What is filtration? Filtration is the process of separating solids from liquids using a filter. What is active transport? Active transport is the movement of molecules against a concentration gradient using energy. What is the difference between endocytosis and exocytosis? Endocytosis is the intake of materials, and exocytosis is the removal of materials from the cell. What is phagocytosis? Phagocytosis is the engulfing of large particles by a cell. What is pinocytosis? Pinocytosis is the uptake of fluids by a cell. What are the types of animal tissues? Epithelial, connective, muscular, and nervous tissues. What are epithelial tissues? Epithelial tissues line body surfaces and cavities. What is the difference between skeletal and smooth muscles? Skeletal muscles are voluntary, while smooth muscles are involuntary. What are the types of plant tissues? Meristematic and permanent tissues. What is the function of xylem? Xylem transports water and minerals in plants. What is phloem? Phloem transports food from leaves to other parts of the plant. What are permanent tissues in plants? Tissues that do not divide and have a fixed structure. What is the function of parenchyma cells? Parenchyma cells store food and help in gas exchange. What are the special features of parenchyma cells? They have thin cell walls and large vacuoles. What is the role of apical meristems? Apical meristems help in the growth of roots and shoots. What is the function of lateral meristems? Lateral meristems help in the growth in diameter of plants. If you found this video helpful and want more short notes on your class books, please like, comment, and subscribe to my channel for more updates.